Good morning, everybody. Distinguished guests, esteemed colleagues, and dear students, a very warm welcome to you all as we celebrate our 15th Foundation Day today. It is not merely a milestone to celebrate, but rather a reflection of the vision that we are turning into reality. It is a moment to honor the collective hard work of our people, our faculty, researchers, including scientists and the young investigators, students, and our staff who have been constantly and continuously pushing their boundaries for the innovation that we are marching towards. As we celebrate our achievements today, we should also remember that we have some future promises to commit ourselves to excellence, make impactful research, and also serve our society. A very happy Foundation Day to you all. We'll begin this by paying our respect to our nation with Vande Mataram. Thank you. I now request our director and all our guests to kindly come forward to inaugurate this function by lighting the lamp. I also request our students to sing the devotional song. I now request our director, Dr. Ravigandham sir, to kindly come. And also I invite Dr. Ramesh, Professor Gauri Shankar sir, Dr. Subir, Professor Ullas, and Professor Uma to kindly come and light the lamps.
वाग्देव्यै च विद्महे सर्वसिद्ध्यै च धीमहि तन्नो वाणी प्रचोदयात्।
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा।
Thank you all once again. May I now request our senior scientist, Scientist H, Dr. Nagendra Hegde, to introduce our chief guest, as well as the guests of honor. I also request our director, Dr. Abhigandham sir, to kindly felicitate them as they are introduced.
नमस्कार, greetings and नमस्कार। हमारे 15वें स्थापना दिवस के हार्दिक शुभकामनाएं। Greetings of our 15th Foundation Day. మా 15వ వ్యవస్థాపక దినోత్సవ శుభాకాంక్షలు। It's a privilege first to welcome Professor Uma Ramakrishnan, who will be delivering a lecture today, a scientist whose scholarship has profoundly shaped temporary approaches to wildlife conservation in India. She is a professor at the National Center for Biological Sciences, Bangalore, where her research integrates molecular biology, population genetics, and evolutionary biology to advance biodiversity conservation. Professor Ramakrishnan is renowned for her pioneering contributions to tiger conservation. She developed and advanced the use of fecal DNA to estimate wild tiger populations through a non-invasive genomic approach, reshaping wildlife monitoring in India. Her research provided insights into tiger movement across fragmented landscapes, identified populations vulnerable to long-term decline, and directly informed conservation strategies. She has also undertaken influential studies on the evolutionary origins of biodiversity in the Western Ghats. In addition, she studies disease ecology, particularly understanding the pathogen dynamics at the human-wildlife interface, an area of increasing significance in an era of emerging infectious diseases and One Health. She has received her PhD from University of California, San Diego, and pursued postdoctoral research at Stanford University, USA, before joining NCBS in 2005. She is a fellow of the Indian National Science Academy, and in 2024 was elected to the American Academy of Arts and Sciences, one of the highest distinctions in international scholarship. Her work demonstrates how fundamental science can serve both knowledge and conservation. Please join me in welcoming Professor Uma Ramakrishnan. We are also honored to have among us Professor Jayaraman Gaurishankar, who is a highly accomplished medical microbiologist known for groundbreaking research on the physiology and genetics of the bacterium Escherichia coli. His research focused on osmoregulation, solute transport, RNA processing and turnover, maintenance of genome integrity, and the interplay between the central dogma, triumvirate, of replication, transcription, and translation. He has authored several original scientific publications and has several patents to his credit. He obtained his MBBS from CMC Vellore and a PhD from University of Melbourne. He was a scientist and group leader at Center for Cellular Molecular Biology and served as director of Center for DNA Fingerprinting Diagnostics for some period. He also served as director additional charge of NIAB. He then served as the director of Indian Institute of Science Education and Research at Mohali. He is currently an INSA honorary professor at University of Hyderabad. He has several accolades to his name. To name a few, INSA Young Scientist Shanti Swarup Bhatnagar Award, CSI Technology Prize, ISCA Millennium Medal, Prakash Basin Award, J.C. Bose National Fellowship, and INSA Senior Scientist Award. He is also a recipient of an international Musilio Schechter Award from the American Society for Microbiology. And in 2013, the government of India conferred on him the Padma Shri for his contributions to the field of science. We welcome Dr. Gauri Shankar. It's our honor to also welcome Dr. Subir S. Majumdar, who molded this campus. Dr. Majumdar is a reproductive biologist and biotechnologist. Over a career spanning several decades, he has played pioneering role in developing innovative methods for transgenesis and advancing the application of genomics to animal and human health. He earned his PhD from the National Institute of Health and Family Welfare. He then pursued postdoctoral research at Southern Illinois University and the University of Pittsburgh, USA. Dr. Majumdar served for many years as a scientist at the National Institute of Immunology, where his lab conducted influential research on male fertility, certainly cell biology, and gene regulation during spermatogenesis. One of his most significant scientific contributions was the development of a non-surgical testis-mediated transgenesis technique, which did not involve conventional embryo manipulation. Along with his colleagues, his work has also contributed to the development of transgenic animals to produce therapeutic proteins in milk and genomic studies for the conservation of indigenous cattle breeds. He served as the director of NIAB for almost a full term before continuing for a short period as a distinguished professor here. Recently, he completed his term as the Director General of Gujarat Biotechnology University. A fellow of all three science academics of India, Dr. Majumdar has received numerous honors, including the Tata Innovation Fellowship and the Labshetwar Award. NIAB welcomes you back, sir. We also welcome Dr. Ullas Koltur Sitaram, a molecular biologist and physiologist whose research has significantly advanced the understanding of metabolism, aging, mitochondrial biology, and epigenetic regulation. He currently serves as director of the Center for DNA Fingerprinting and Diagnostics, and he has also held additional responsibilities at NIAB. He is also a professor at the Tata Institute of Fundamental Research. He has contributed fundamental insights into how cellular energetics and inter-organ communication influence aging, cancer, neurodegeneration, metabolic diseases such as obesity and diabetes, stress adaptation, and organismal survival. Beyond fundamental biology, he has contributed to important translational initiatives. During the COVID-19 pandemic, he played a leading role in conducting one of India's largest serological surveys. He has also recently initiated collaborative interdisciplinary programs on malnutrition, non-communicable diseases, metabolism, aging, linking basic biology to clinical science. Professor Koltur Sitaram's scientific contributions have earned widespread recognition. He is an elected fellow of all three science academies. Among several honors, he has received the Swarn Jayanti and J.C. Bose fellowships in recognition of his outstanding scientific achievements. Welcome, Dr. Ullas Koltur Sitaram. And finally, we are deeply honored by the presence of our chief guest, a distinguished officer of the Indian Police Service, whose career has been marked by exemplary public service, institutional leadership, and a commitment to the rule of law. Dr. M. Ramesh is a 2005 batch IPS officer and presently serves as the Commissioner of Police, Cyberabad. Over the course of more than 2 decades in service, Dr. Ramesh has held a series of important operational and senior administrative assignments within the Telangana State Police, including being the Inspector General of Police for provisioning and logistics. As Commissioner of Police, Dr. Ramesh has brought the depth of his experience to one of the defining security challenges of our age, that's cybercrime. Under his leadership, Cyberabad Police has intensified coordination with the banking and financial sectors, established round-the-clock cybercrime response mechanisms, and expanded public outreach initiatives addressing digital fraud, financial scams, and emerging AI-enabled threats. Dr. Ramesh holds a BA honors degree from Kirorimal College, Delhi University, MBA in Technology Management, and PhD in Law from Osmania University. His thesis focused on legal and criminological studies. His contributions to public service have been recognized through the award of Police Medal for Meritorious Service, conferred in recognition of distinguished and meritorious duty. We are privileged to have among us a leader of such distinction and public purpose. Please join me in welcoming our chief guest, Dr. M. Ramesh, Commissioner of Police, Cyberabad. Thank you.
Thanks, Dr. Ravi. Now I welcome you on stage to address the gathering.
Good morning, all, and Hare Krishna. Respected dignitaries, honored guests, esteemed colleagues, and dear students. At the outset, let me take a small quote from Panchatantra, which marks this Foundation Day. Udyamanahi siddhyanti karyana manorataha. The meaning is, like, success is achieved through efforts and not by mere desire or daydreaming. Let's start our day with this small verse, and it is with this spirit of dedication, perseverance, and purposeful action that we gather today to celebrate the 15th Foundation Day of our institute, a premier institute shaping the future of animal biotechnology in our nation. It is a privilege to address this august gathering on this occasion. Foundation Day is not just about marking time. Mark my words, it is about measuring transformation. This day reminds us of the foresight of our founders who envisioned an institute dedicated to advanced animal biotechnology. I repeat again, the Foundation Day reminds the foresight of our founders who envisioned an institute dedicated to advancing animal biotechnology for national development and scientific excellence. From its inception, NIAB has stood as a beacon of innovation, interdisciplinary research, and societal impact, contributing significantly to livestock health, productivity, and sustainability. This is the time we celebrate our achievements, express our gratitude to our pillars. When I mean pillars, leaders and policymakers, directors and scientists, administrative team, technical staff, outsourcing guys, secretarial staff, and as well as security staff, and finally our students who are the future torchbearers of science. It is your collective effort that transforms vision into reality. This vision has evolved into a dynamic ecosystem where molecular biology meets data science and fundamental research meets translational impact. Today, NIAB stands at the forefront of cutting-edge research areas such as functional genomics and genome editing, reproductive biotechnology, computational biology and genomics, including bioinformatics, next-generation vaccines and diagnostics, and stem cell research. These domains position us strongly to address emerging challenges in food security, climate resilience, animal health, and productivity. As we look ahead, our responsibility becomes even greater. We must strive to foster innovation, entrepreneurship, and interdisciplinary collaboration. Dear students and young researchers, science is not just a profession, it is a commitment to truth, curiosity, and humanity. Let's take a leaf from 2.47 Bhagavad Gita, Karmanyeva adhikaraste ma phaleshu kadachana. What does that mean? This will guide us in your work and will let us know that we'll have to work with dedication and integrity without being bound by immediate outcomes. That means like our efforts should go on and go on. Finally, as we celebrate our Foundation Day, let us affirm our commitment to excellence in research, integrity in action, and service to society. Let us ensure that our science remains ethical, inclusive, and impact-driven. Let us move forward with this renewed energy to build an institution that not only excels in science, but would transform lives. Thank you, and Jai Hind.
Thank you very much, sir, for the powerful message. I'm sure under your leadership, we'll be achieving those, an impactful research for serving the society. May I now welcome Dr. Ramesh to address the gathering.
Good morning to everyone. The director, Mr. Ravi Kumar, Professor Ramakrishnan, who is the main speaker for the day, Professor Ullas Kultur Sitaram, Professor Subir Mazumdar, Dr. Gauri Shankar, all the research scholars, academicians, and friends. It gives me immense privilege, rather goosebumps, to be amongst the scientific community. In fact, I love science, but somehow I've become a student of arts. I don't know how. I was interested in arts up to my high school, in sciences. Somehow the path changed. Even now I'm very much interested in science. Anything that comes, I would like to know, learn much about it. But somehow my traction changed and I got interested in the art side. Later on, towards the end of this career, I mean, I have finished 30 years of policing. Last 5 years, I was into research. I always believe that I will be continue to be a student, learn more and more new things. And coming here for the first time, my friend and my classmate, Ravi, brought me here. We both studied together. Your director and me, we both studied together. That's how I'm here. Otherwise, a commissioner of police wearing a khaki wouldn't be in the precincts of any learning institution. Somehow, he came personally to my office, told me about this institution. And in fact, I was very much overawed by the moment I entered into the campus, such a beautiful thing. I asked the administrator, Sorry, I mean, I do not know your designation, sir. Senior manager from the administration. I asked him, such a beautiful campus, how long did it take? He said, 4 years. Wonderful. Wonderful. It's unimaginable. Such an institution coming with entire landscape and in the 100 acres development in this area. That's really, and he also told me how he managed and got this done. Wonderful, sir. Congratulations to you. And coming back to animal biotechnology, while I was working as DCP in Hyderabad city way back during the COVID times, I had an occasion to visit National Institute of Nutrition, NIN in Tarnaka. This also belongs to the same ministry, maybe food and agriculture or science and technology. ICMR, okay, yeah. So I had an occasion to visit their labs, seen what's happening there. Then this is the second time that I'm coming into a research institution, place of higher learning. I'm very, very proud of you all. You all remember how the vaccine got generated in Hyderabad, and especially in the industrial area in Cyberabad limits, we have so many pharma units, biotechnology units, which are contributing to enormous foreign reserves for the country. We are earning a lot of foreign reserves, despite our imports of oil, despite depletion of our foreign reserves due to various other imports. Our technology has come to the rescue of our country. And I had a classmate who is in the top position in one of the pharma companies in the US. It's based in Hyderabad also. He comes and he told me, in one police station area, in Pasha Mailaram in our area, 30% of American injectables in North America, 30% of entire North American injectables are produced in one place. I got literally scared. I mean, such a vital installation, and we don't know. Because we as police are supposed to know what is a vital installation, what is the value of that, and then value of each and every scientist. Even their life is a threat. There is so much of competition in the entire world to take away the patents. Every week almost we receive one theft, intellectual property theft in various research institutions, pharma companies and all. That's also a big threat. If you're not able to divulge that information, they may even cause physical harm. So these things we need to know. And coming here inside this campus, this is also equally threatened. Most of you are dealing with very, very, very much vital information, and then which has a potential of bringing a lot of foreign reserves into the country. When you make vaccines, and that just like our Bharat Biotech did, just like what other Poonawalla did, so we have that potential inside our country. And this research is aiming at not only these animals, even all kinds of animals which aid in our country, in our agriculture, milch animals, cattle, so much. I'm not going into the details. I'm not really aware. Please don't find fault with me. But this kind of research aids in agriculture as well. When livestock development takes place, a lot of gene mutations are happening in CCMB. I'm not sure whether they're doing it in the CCMB or not. But so much is happening. We have seen dwarfed cattle also in the recent days. I don't know how it's happening, but somehow I don't know whether it's natural or unnatural. Things are happening that way, and scientific community should come out with the answers. I don't know what is the benefit out of it. And while we were studying in the schools, we only used to learn about high yielding varieties, which brought in green revolution into the country. But then manipulating with the life, with the biotechnology, creating new beings, it's all we used to see in Hollywood movies. Now, in front of our eyes, things are happening. And I hope and trust that this institution on the 15th anniversary takes a firm resolution that we all will try and contribute to the knowledge of our country, which will take to the next heights and compete with all the developing countries. No, we are actually competing and they're threatened. Most of the foreign countries are threatened with us. And I am hopeful that you young scientists will definitely take India to the #1 position in the country, and we will be able to reach all the Millennium Development Goals by 2030, Sustainable Millennium Development Goals, as well as the 2047, 100 years of Indian independence, what the government has resolved and kept as a target for 2047. So all those targets we would be able to reach in the next 20-25 years. So with this, I would like to conclude my address, because if I speak more, I would appear to be very ignorant about these scientific things. And having come over here, I would like to come more frequently to learn developments about this, so that at least I learn something, and then when I go somewhere else, I can speak something what I know. So I request Ravi to invite me sometimes to learn something new, some new innovations. I would not hesitate to come despite my schedule outside. I would like to come and spend some time with the scholars, so that I'd like to learn. Thank you so much. Jai Hind.
Thank you very much, sir. We are honored to have you here today. May I now request Professor Gauri Shankar, sir, to share a few words of wisdom.
Yeah. Okay. Yeah. Good morning, everyone, and my congratulations to Dr. Ravi and all the faculty, students, and staff of the institute as we celebrate the 15th Foundation Day today of this NIAB. I didn't expect to be called to speak. I was just going to be a silent spectator, and I was looking forward to Uma's lecture, which was going to follow. At this time, so it's easy for someone like me to go into the past, which I'll try to keep as brief as possible, but spend a little more time about the future of this institute. So as far as the past is concerned, it looks like the founder director not being here, I'm actually standing in his stead because I was put in charge of this institute immediately after his term was completed, Dr. Reddanna. The institute owes a lot to the founder director, Dr. Reddanna, for having conceived the the format of this place, and in putting it together in terms of hiring people, getting the land, which is very, very difficult in a place like Hyderabad, and initiating the process for setting up the campus, right? So we can look at the first 15 years, which has basically been completed today, let us say, as the setting up phase of the institute. Now that I think all of us must realize is almost completed, in a curve that we can talk of in biochemistry as a as a hyperbolic curve. We are more than 95% completed. The building is there, the infrastructure is there, the faculty have been recruited, and there are a very large number of students, which of course the numbers should go up, but that's important. So I think what one expects from the country's point of view is for the institute to to start in what I would call a massive productive phase as we go along, right? And for that, it is well set in terms of the resources that it has already developed. If we look at the future, I just wanted to convey one point, which I happened to speak about in a talk to veterinarians about a few months ago. And since the mandate of the National Institute of Animal Biotechnology is very, very closely related to the veterinary aspects of health and welfare, right? we all have heard of the phrase One Health, right? And One Health is something that has become very popular as a way of bringing people together in the last, what I would say, 10 or 15 years. One Health has classically been taken to represent two aspects of research or work. One are the zoonotic infections, the zoonosis of infections of parasites, viruses, bacteria that can transfer from animals to humans, and therefore it is a One Health concept. And the other is this increasing awareness and problem of antimicrobial resistance, which again transcends the boundaries of the animal kingdom and human health. What message I wanted to say, which is appropriate for the faculty and the students of NIAB, is that let us look at One Health as an even larger concept. And I'm very happy that actually Professor Uma is here in this audience today. So One Health, of course, covers animals, it covers humans, but it covers the environment as well in what sense. So that is something that is already being recognized. But I also want to say that we can look at One Health as something which covers the interplay between physicians and veterinarians in all aspects. If we look at it that way, what else will come into the world or universe of One Health? We can look at transgenic animals which are used for biotherapeutic production. That is also, somebody has to do that. And that could well be one of the research mandates of this institute. Transgenic animals for better nutrition of the human population, which could be in the form of, of what is already going on, improved milk production, hypoallergenic milk, or the double meat or the double muscle animals, and so on. More than that, now we are also talking of xenotransplantation. So that is something that I would think is part of a One Health concept, because we are now looking at organs being transplanted from farm animals like pigs into humans. Already there are substantial success stories which are only going to become better as we go along. And then we have additional examples of the One Health concept in the way that I am defining it. I don't know how many of you have heard of the vulture population being decimated in a very, very indirect way, that farm animals are treated with an analgesic called diclofenac, and that for some reason can be very, very toxic to vultures that feed on animal carcasses. So somebody, so these are the kinds of aspects that, if you look at it, cross the boundaries of what we can call as the verticals of animal welfare, human welfare, or environmental welfare. And we are talking of interdisciplinary problems. So I thought with that, I will stop here, say that, the past has been great for the Institute, but let us look forward to a very prosperous and productive future for this Institute. All the best to us.
Thank you very much, sir, for the insightful thought, and we'll definitely march towards that as well. May I now request Dr. Subir Mazumdar to add more to the impact that has already gained momentum right now.
So good afternoon. Can you hear me?
So,
I think it's afternoon, and it's my pleasure to be back here. And I first of all congratulate and give my wishes to the whole institution for the Foundation Day. And while lighting the lamp, I was remembering that we celebrate actually 2 Foundation Days. One when it was conceptualized, this is today, and on 11th of August.
11th.
August, coming 11th August, it will be 10 years that we were lighting lamp here, all of us, with Harshvardhanji to inaugurate this building. So earlier we were in a temporary location, and the building work started. And the first thing being coming from an institution which is created by a very big giant of the biotechnology of this country and internationally known, Professor G.P. Talwar, I looked into the aesthetics of this, and while sitting in Miyapur, first thing we did is put Anand as in charge of being here every day and implant as many trees as possible. So the trees started growing before the building grew. And today that's the outcome of that beauty we have. This was completely a hilly area. Nothing was there. Whenever you touch, it was a crusher area where they used to crush stones for the roads and buildings. Dust of crush was all. So at least 5-6 inches of layer was that. But I am grateful to God, as it's said that if you put a lot of labor, I think intelligent people are there in this world. And what I found, I am not intelligent, my IQ is very less, but I thought everyone who works very hard, guaranteed success comes. So I am giving an example that if all of you can work hard, even in such a stony area, you can grow such a wonderful institution. So I'm grateful to Harjit. I'm grateful to Professor Redanna, who actually, the red carpet which you are coming in, he is the person who made this red carpet entry for all of us to come and work to build this wonderful institution. So these were 2 classrooms. So we broke it. Half was done for separation. I said, government will never give money to make a auditorium. But this is illegal. I said, let it be illegal. Let us make an auditorium. Let us go back. The government will never give money for making an auditorium in future. Whatever money you got, you got. So we got a permission, got converted. Our new stamped sanction for this place came in. So I am grateful to God that we have such a wonderful auditorium, because in system I worked for a long time and I know how government works. The institute is full of great instruments. It's full of great scientists. When I joined in Miyapur as a director, there were only 6 scientists. Sarvar Ajam, kaha ho kya tum? As usual, he is absent. So, yeah. So there were 4 PhD scientists and 1 Sarvar Ajam who was Masters of Bioinformatics from JNU. And I'm very impressed that you went from here to St. Stephen's too, because I spent 32 years in Delhi. So when you talk about the Delhi institutions, I know how much studious you would have been to get entry into St. Stephen's. That I know very well. And you may have a lot of other interests also to go there. So with that 5, for all 5 of them, I was the person who interviewed them. Professor Gauri Shankar was also involved in many recruitments here. And I have also made him chairman of several committees which recruited people. And from 5, the strength has now increased to how much? Huh? But ज़्यादा था, कम हो गया लग रहा. So when I left, I think it was around 26. And we selected all right people to do good research. And those who were not right, I fought with them, misbehaved with them for science and made them good people to do good research. And so this institute is now a vibrant institute. We were all in the frame of work of extremely basic science, but gradually we converted the philosophy that government wants something to be product oriented, something for the society to be handed over. And at least more than 60% of them have started thinking that way. Therefore, the growth of this institute has occurred very nicely. All the people here are wonderful. I have a lot of memories of this institution, good and bad both. And I think, but all of their end point was growth of the institution. So remember, And I thank all of them who were there at my time, that they really worked very hard with me day and night, all Saturdays and Sundays, to make a culture of the institute that we have to work hard to become one of the fastest growing institution at that time. And I think I'm very, very happy that everybody takes pride in that, and they continue to do work in the same fashion as they used to do. To the students, I usually, now I start getting very emotional. So I built institutions and forget about it. Because while you go, you need not be necessarily remembered by the institute for a long time. पर जैसे माँ की इच्छा होती है कि बेटा मुझे हरदम याद रखे, हर दिन फोन करे, नहीं हो पाता है. ऐसी ही मैंने मेरी इच्छाएं माँ वाली छोड़ दी है और अभी मैं जो है दूसरे इंस्टीट्यूट में चला गया. So that has become one of the fastest growing institute and university in the country, Gujarat Biotechnology University, mind-boggling institute. So you will be, if you are scientist, you will know that in 2.5 years of writing grants, we received 70 extramural grants. January to March, 17 grants, 11 DBT, 3 NRF, 1 Gates Foundation. I am now worried. It's very difficult to contain success. Getting success is something. So now I just retired 3 days ago from there. I'll forget that institute. It's quite possible. I don't want to say कि मैंने ये किया, मैंने वो किया. I want to say हम सब ने इंस्टीट्यूट बनाया है. हमारा सबका इंस्टीट्यूट बहुत अच्छा है. हम सब ने गुजरात में इंस्टीट्यूट बनाया है. हमारा वहां पे इंस्टीट्यूट अच्छा है और मैं उसको भी भूल जाता हूँ. And it is quite possible कि जैसे शादी करके बेटी घर छोड़ के ससुराल जाती है, मेरे साथ थोड़ा उल्टा हुआ है. बेटियों की शादी करवा कर मैं मां हूं. बहुत हद तक मैंने इस इंस्टीट्यूट को फीड किया है, मिल्किंग करी है, मिल्क दिया है. और इस बेटी को यहाँ छोड़कर मैं चला गया गुजरात. वहां एक और बेटी बड़ी करी है. बड़ा इमोशनल रिलेशन है. आपको समझेगा जो इंस्टीट्यूट बनाता है कितने तकलीफों से जाता है. रात रात भर सोते नहीं थे. कई लाइसेंस दिस इंस्टीट्यूट वाज कैंसल्ड. डू यू नो दैट? बिकॉज इट इज नियर दिस लेक. And CDFD was cancelled on paper where they went up to Supreme Court and they said nothing doing. Lake, it is less than 10 kilometers. So you're gone. In our file, it was written like CDFD, this institute is also within the 10 kilometers, it's cancelled. So Harjeet knows and I know how we made it clear that we got it cleared for making an institution. That's a great thing. Wonderful. तो एक माँ के हिसाब से मैं इंस्टीट्यूट छोड़ के गुजरात चला गया. गुजरात में और बहुत बढ़िया इंस्टीट्यूट बनाया. कभी इंटरनेट पे सर्च कीजिए गुजरात बायोटेक्नोलॉजी यूनिवर्सिटी, आपके समझ में आएगा वो क्या चीज है. ओनली 3.5 years old इंस्टीट्यूट. और जैसे बेटियां चली जाती है ससुराल शादी के बाद, तो मैं वो माँ हूं जो 2 शादियां करा के मैं मैके जा रहा हूं दिल्ली. बिकॉज 32 years I was in Delhi. I did my PhD in Delhi. Then I was for 4.5 years in USA, but then I again worked for 22 years in Delhi. So in touch wood, in all probability, this is not wood. This is wood. I will be probably going back to my Maika, which is a place in Delhi. तो ये उल्टा हुआ कि माँ बेटी को ब्याह के अपने मायके चली गई. So why I asked the audience to be with me indirectly, and why I have made you laugh, that I want that this vibrations of laughter and happiness should always be here. Dukh har kisi ke saath hai. Pain and suffering is part of life. Forget as long as you're alive. I used to teach the all PhD students. PhD कर रहे हो 5 साल रहोगे. Simple criteria है. पहले से जान के रखो. पर जिंदगी उसके बाद बहुत हंसी खुशी और मस्ती की है. Guaranteed रोना है. Up and down होते रहेगा. Similarly, I am saying कुछ भी अवस्था हो, keep smiling like this. Always be happy. And so that there are good vibrations here. And I'm proud of my administrative people here, my daily wagers here, my electric people here, air conditioners here, my faculty here, and all the students here, that they are part and parcel of. They all together, my guards here, who have made such a wonderful institute as a family. So stay like a family. I will wish all of you for that. And with this, I thank Professor Gauri Shankar, Ullas, Madam, for coming here, including Ramesh Saab, and it's nice of his. And everyone knows Ravi is very close to my heart, and he has no choice than doing good for the institute. Anytime he does, I will never come to ask for a penny, ask for a favor or a cup of tea to him. Never. But I will remain far away. Whenever he is about to commit a mistake, I will hold his leg or his hand. That promise I do for him, but I believe in him. That is a wonderful human being, wonderful scientist, a great thinker. And very rarely in this age of time, 2025 or 6, whatever, you get a person of that kind of frame of mind. So you are all very, very lucky to have him here. And I am grateful to God that I am very sure that he will take care of all of you. With this, I will close. Thank you very much.
Thank you, sir. The building of this institute under your guidance is now depicted in terms of the beauty and also the science that the institute. Thank you. Now we'll have the pleasure listening to Professor Ullas. It's our pleasure, sir. Please.
First of all, I would like to congratulate all of us because I still believe that I'm part of NIAB. Although I was here only for 8 months, it actually feels like homecoming. So I think many things have been said already. NIAB has great potential, of course, I think all of us know this. While we are very happy congratulating ourselves, I think we should also be aware of the demands that the Government of India and the society has placed on us. This is one of those unique institutions which is unlike any other in the country. It's not an IVRI, it's not a veterinary research institution, it's not a regular biotechnology institution, it's an animal biotechnology institution. So the kind of things that we will do here, I don't think will be matched by anything that happens anywhere else. So therefore, I think the mandate is challenging, but also that's a unique opportunity. And we have a fantastic leader now with Ravi being in charge, with Subir holding his leg. I don't think any of us have to worry. We can all relax. The most important thing is, at least I think you've heard me many times when I was here for a short period. I still feel that there is a huge opportunity to connect with the industry on one hand, and other connect with the veterinarians and field, looking at field aspects. Otherwise, doing lab science can be very self-fulfilling, and we may have great things going on, but unless we also make effort, especially given the mandate of this institution, I think we'll be failing in our efforts. I think all of us know that, we're also a part of this new organization called BRIC. I encourage all of us to look at the options of collaborating between institutions, because although nobody stopped us from collaborating with each of the other DBT institutions or elsewhere, with this BRIC, there is new momentum, new opportunities, and I think we should make utilization of that. And I'll really encourage Ravi to reach out to all the different organizations within the DBT system to do something which individual institutions cannot do. Of course, I can't be as humorous as Subir is. He's one of the most engaging and entertaining person I've ever known in science. I'm sure you've listened to his songs. Otherwise, you can call him back and then ask him to sing. I think NIAB also has this unique distinction of having too many directors with very different philosophies. I empathize with you. But just the fact that you are able to bear with us means that you'll do well. So on that note, I'll stop, and then congratulations to all of you again.
Thank you very much, sir. Now we'll have national anthem. Thank you.
Vinayaka Jaya भारत भाग्य विधाता पंजाब सिंधु गुजरात द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Before we break for the high tea. And after the high tea, please assemble back. We'll have the talk by Professor Uma.
This fire only, madam? Same fashion, this way? Same.
started in doing science because of this interest and curiosity about animals. And what's really amazing is that, we live in India, a very small country considering global land area, but with a disproportionately high proportion of biodiversity. And what's really amazing is that, around us are all of these species. This is the hard-ground barasingha, this is a leopard. They've evolved over millions of years, and this diversity surrounds us. And we're always curious about it. We watch documentaries, we watch National Geographic, and we wonder, though, how can we understand more about them? How can we deconstruct the mysteries that surround us in nature? And I've basically used genomics as a tool to do this, and I'll talk you through some of the stories of the work we've done, but also focused mainly on tigers, a little bit of work on other species, some applications of genomic tools. More recently, been working in the area of disease ecology and evolutionary dynamics to understand spillover of potential pathogens. And then maybe in the end, because this is a foundation day, I'll spend a few minutes, if I have time, thinking about, together with you, the possible future. So, biodiversity surrounds us, and it's also critically important to us. The air we breathe, the fruits we eat, the water we drink, all of it comes from biodiversity. However rich the nation, greater the technology, we've not been able to manufacture water, right? So especially in India, even pollination is mostly by wild insects. So a lot of everything that we depend on actually comes to us from nature. So while we may debate it, biodiversity is critical in India, and even more so in the perspective of livelihoods in India. A lot of people in India depend on nature for their livelihoods. At the same time, biodiversity is a threat. So you might have heard or seen these kinds of statistics. Almost 1 in 2 amphibians is threatened with extinction. 1 in 4 mammals is threatened with extinction. And this is crazy to think about, right? All of this biodiversity that surrounds us, supports us, sustains us, is actually potentially at risk of extinction. And of course, concomitant with this, so actually, if you look at the data, it's since 1972 that the dramatic declines in biodiversity actually have started, population extinction, species extinctions. But concomitant with that, there's been an amazing rise in technologies for sequencing. You all know this. For example, it's quite sobering to think that the structure of DNA, for example, was not even discovered 100 years ago, right? And the ability to sequence DNA, pioneered by Sanger, was actually very, very recent. But it was basically PCR and the next generation sequencing, which resulted in a massive change in these technologies. So basically, next generation sequencing broke Moore's law. The first human genome sequenced cost $10 million and 10 years, many, many labs working together. But today, you can actually sequence a human genome for less than $500 and in a few hours, right? So clearly, this technology has exploded, and hopefully, these two things next to each other, the decline of biodiversity and sequencing technologies becoming cheaper, allow us to study it better. But what actually makes populations go extinct? We're all biologists here. And what I've told you so far is more like what you read in the newspaper, more sensationalist media stuff, right? So as we've studied populations, we know that small populations, because of stochastic effects like inbreeding, genetic drift, demographic stochasticity, changes in birth rates and death rates, environmental stochasticity, have high probabilities of extinction. So for example, you can see here, when the population size is small, extinction probability is high, but very soon this curve decays. So even populations of 100 or so have appreciably small extinction probabilities. The other thing we do know is that when there are a few isolated populations, the probabilities of extinction are high. This is number of patches versus time to extinction. Whereas many connected populations, many populations which are distributed in space with connections, like a network, if you will, have low probabilities of extinction. From a perspective of genetics, again, these are recently redone data. So it looks like the bottom part is getting cut off. But basically what this shows is, for example, that when populations have low genetic diversity, for example, these populations, low heterozygosity, they have high chances of extinction when genetic variation itself is low. And this is generally because small populations have low genetic variation, and small populations are subject to what's called genetic drift, or the chance fixation of alleles, which could be good or bad. And also you have basically inbreeding and inbreeding depression. Inbreeding is mating between relatives. Inbreeding depression is the exposing of recessive deleterious mutations because of inbreeding, leading to decreased fitness and survival. So overall population extinction depends on population size, population isolation, and genetic variation. Now, if you think of the narratives of conservation, you see, I'm sure all of you have read in the newspaper about tiger numbers are increasing, right? But all of those narratives are just focused on numbers. They're not necessarily focusing on population isolation or connectivity or, for example, genetic variation, despite the fact that recently the Convention for Biological Diversity has explicitly incorporated sustaining genetic variation as a guideline, because even our crops, even our domesticated livestock, if we lose the wild counterparts, we will be losing potential genetic resource. So what about India? Is really something we should be worrying about in India? Well, these pictures are really, really powerful. They're basically projections of what India was like in 1700, and this is almost 25 years ago now. And you can see that what was, forest, rangeland, grassland, and so on, has now become primarily agriculture and villages, right? So basically, India has gone from 60% forest cover to 9% forest cover, and basically villages and agriculture have increased exponentially. So imagine now that your wildlife, say a tiger or something else, which lives in these small pockets. You're basically like a species stranded on an island amongst a sea of humans, right? So you're effectively isolated. Populations might be small, and that's what basically we're dealing with in the context of wildlife. So I basically use tigers to try and understand extinction. And this is a good system to understand extinction because tigers have already seen extinctions in the Caspian region, the South China region, island Southeast Asia. Tiger populations have already gone extinct. And this is not a very sensitive species which lives in a very small location. Look at that, living all the way from, Russian Far East and tundra to tropical rainforest. A very, very adaptable species, a very widely distributed species. Yet today, only about 4,500 to 5,000 tigers exist in the whole world. Okay, so you can imagine tigers have pretty much lost 95% of their range. And while we are celebrating tiger recovery in India, the fact remains that globally, tigers are still in quite a lot of danger. So the questions we are interested in is, will tigers go extinct in India? Why, where, and can we actually do anything about it? So basically, like I said, we use genomic information for this, and you all know very well what a genome is. Just to remind you again that, often we look at markers, we look at specific bits of DNA regions, right? And these markers actually end up having low information content, whereas you can think of the genome as a large puzzle, and basically the information in the genome has positional information as well as something about content. Like, for example, this is the back of the snow leopard. So you have that information in this piece. So basically, using the whole genome gives you much richer information content than if you use markers alone. Okay. So basically, in order to work on tigers and work on genomic lenses to study tigers, we had to first create a reference assembly for Indian tigers, which we did. So there was a tiger genome assembly which was published way back in 2013. This was from a captive Amur tiger or a Russian Far East tiger. But basically in 2023, we created an assembly for Indian tigers. The tiger genome is similar to the human genome, 2,400 million letters long. And this now serves as a baseline against which we can map all tiger genomes to look at how variable or not variable they are. Okay, so when we did this, the first thing we wanted to understand is how variable are Indian tigers vis-a-vis tigers across the world. So we looked at range-wide population structure. So I'm a population geneticist. So, all the stuff I'm going to show you is about gene flow and genetic variation and so on and so forth. And so basically we were able to sample tigers, wild tigers from across India, some from the Russian Far East, some from Malaysia, and then some first generation captives from Indonesia. We were not able to sample tigers in Thailand. There are very, very few individuals there. But when we sequenced these genomes and we looked at, first of all, you always look at population differentiation. You can't compare apples and oranges. If I want to compare genetic variation of Indian tigers to those in Indonesia, I need to know whether they are separate or the same population, right? So basically this PCA shows you, first of all, variation is not very high. So these tigers are not very differentiated from each other. But you can see, for example, that Bengal or Indian tigers are one cluster, and the so-called subspecies, Sumatran, Malayan, and East Siberia, which is Amur, are separate clusters. What's really interesting is this plot at the bottom here. It's a rarefaction curve because we have different sample sizes for these different subpopulations, populations. When we look at genetic variation or the number of alleles per locus, how does that change as a function of sample size? So what you can see here is the pink, the hot pink is Indian tigers, and basically they have disproportionately high variation. So even when you have 20 Indian tigers genomes, you're not saturating in terms of variation. So in fact, it turns out that Indian tigers have more than 70% of global genetic diversity. Okay, so that's really interesting, and it's also important because it kind of gives more, weightage for conservation in India. But I'll also point this plot out. So this is an admixture plot. You can see, for example, these three individuals have bits of green and this like burnt orange, and that is because they are from here. They're from Kaziranga. So you can see that while there are these distinct populations, there's signatures of what was a long and continuous population. So I'll just tell you a couple of stories. I won't talk about this work. Basically, we looked at connected populations in central India, but I'll tell you these two stories about small and isolated populations and what happens to them. This is important in the context also of livestock. There's a lot of inbreeding and selective breeding for livestock, and this will result in similar inbreeding effects as we see in these wild populations. So I already explained what happens when you have inbreeding. Imagine if you have these deleterious mutations in a population, they're usually carried as recessives. They're not selected out because as recessives, they don't have any effect. And so they kind of hide in populations for very long periods of time. But what happens when you have inbreeding is individuals who are related have the same recessives, right? The same deleterious recessives because of shared ancestry. And when they mate, they do have offspring which carry both copies or carry unmasked recessives. So now you have two recessives coming together, and this individual may be diseased or for some reason have lower fitness. This is called inbreeding, and this is called inbreeding depression. So we basically expect higher recessive load as well as higher frequency of recessive alleles in inbred populations. So we tested this in the context of tigers in India. We sequenced genomes from 3 kind of subpopulations in India, Rajasthan or Ranthambore, where tigers are isolated. There's a small population, the central Indian tigers, which are well connected, a 2-dimensional population, and tigers from the Western Ghats, again connected, but slightly less connected than in central India. When we sequenced several genomes at high depth from these individuals, what we found was really interesting. This is a plot of what's called runs of homozygosity. This is basically what proportion of the genome is in stretches which have no variation at all. And F is the inbreeding coefficient, which can be estimated from the proportion of the genome in runs of homozygosity. And what you can see is, for example, for this small isolated population, almost 40% of the tiger genomes have 40% of their genomes are homozygous. Okay, so this is really, this is pretty high. For example, if you see wild populations, inbreeding coefficients of higher than 0.2, right, do cause fitness effects. On the other hand, look at central India, the same species, right, very close by, like a few hundreds of kilometers, but because these populations are potentially connected, you see low proportion of the genome in homozygosity. Okay, so what this showed us was that small and isolated populations, and how long has Rajasthan been isolated? Not very long, maybe, I don't know, 50 years or something like that, maximum. They've also been through a population bottleneck when the tigers crashed, right? So in 1972 was when the Wildlife Protection Act came into place, as well as the Project Tiger started, and that's when protection of tigers started. Prior to that, it was a free-for-all, hunting, etc. was all going on. And so maybe now we have 4,000 or so, 3,000 something. There may have been much fewer at that time. So these old bottlenecks, as well as the subsequent isolation, has resulted in tigers here being twice as inbred as those elsewhere. So that's nice to know that they are inbred, but that is coming from a genomic metric, right? That is, we sequence genomes and look for these signatures of inbreeding. But inbreeding is mating between relatives. So can we observe inbreeding in the wild? Okay, this is really tough. Again, a great advantage you guys have with livestock, because you know the pedigree, right? So you have these massive pedigrees, and with pedigrees you can do fantastic analyses on fitness effects of deleterious mutations. Why? Because, for example, if you have a deleterious mutation, there would be what's called segregation distortion. It will not be inherited in a Mendelian frequency, because maybe it's lethal in the embryo and so on and so forth. So you can actually look at functional effects of deleterious mutations. That is the holy grail. Today, while we've sequenced thousands and millions of genomes, our ability to connect genotype to phenotype still remains weak. And we cannot do functional studies for every single mutation, right? So basically, we wanted to see whether we can actually reconstruct genomes in the wild. So this is a crazy project. We've been doing this for 10 years now, where we follow individuals. So for example, we follow this tiger. This tiger is sitting here. The forest department maintains a register of all the individuals identified by their stripes. So we identify the individual, cross-check it with their ID. And once this individual gets up and walks away, we find a lot of shed hair of that individual sitting over there. And with these few, like 20, 30 hair, we are able to sequence genomes. Okay, so now I can try to get blood samples or tissue samples from individuals, but those will only be individuals the department is trying to collar or move or something like that. But this way I can control what samples I get. And then I can basically test for paternity and reconstruct a pedigree. So this is what we've been doing for the last 10 years, and we continue to do. We have now about 70 genomes, and we have about 25 mother offspring pairs sampled over 5 generations. They have overlapping generations. And what we find is, very interestingly, for example, there are only 2 mitochondrial haplotypes. So all the tigers, there are 90 tigers today, but all of them trace their ancestry to 2 females, right? So it was thought, or if you look at the records, foresters suggest that there were like 9 families which started Ranthambore and so on. 9 is already a small number, but our data suggests there's only 2 mitochondrial haplotypes. This is the reconstructed pedigree, and you can see all of these loops. Those loops are basically signifying inbreeding, right? And what you can also see is that further down, so basically, further down in the pedigree, this is more recent, there is more of this red, sort of. The reds are matings between relatives. So what we're seeing is, in the recent times, there is more mating between relatives. There is no first degree relative mating. That is, we don't see matings between mother and offspring or sibs, but we do see second degree relative matings. How has this impacted genomic inbreeding? So basically, this is a plot of that same FROH, the inbreeding coefficient as estimated from runs of homozygosity in the genome and versus birth year of individual. And you can see that, remember I was exclaiming that 0.4 was already quite high. That is 40% of the genome being homozygous was high. Now, for individuals born relatively recently, almost 60% of their genome is homozygous. So these tigers, I mean, it's kind of obvious. They're isolated. They are mating within this small reserve. And we can see in real time that the inbreeding and inbreeding coefficients are increasing. So the questions, of course, are still many. Does this impact survival? Does it impact their fitness? And we're trying to look at this with multiple ways. Of course, one way is to look at reproductive success over time, but our sample size is very small. So we're also trying to look at, for example, there are some females who have never reproduced, or we don't know whether they had cubs and the cubs died or whatever, right? They don't seem to have reproduced. We're trying to look at endoparasites, hormone levels, behavioral differences, various approaches to try and understand whether there are some fitness consequences. Now, inbreeding may not necessarily impact only fitness. It could also cause deformities, for example, right? So there are some individuals that have crossed eyes, again, a very common phenotype in inbred white tigers, for example, what's called strabismus, which is crossed eyes. But of course, these are complex traits. We're not sure whether we'll be able to figure it out as to what is the genetic basis for it and what particular deleterious mutations to look out for, but we do want to try and understand this better. So this is very quickly, I'll go through this other really fun story. This is the story of this mysterious black tiger from a forest in Odisha called Simlipal. So the locals who lived here for a long time talked about a black tiger, and nobody believed them. They said, oh, you're lying, you're making it up, and so on and so forth. Until in 1993, a youth in self-defense shot a tiger, which is basically black, right? And then once camera trapping started, these pictures started surfacing. Now, this is not a melanistic individual. This is not like a black leopard, which is completely black, right? Tigers have three colors, orange, white, and black. And this individual has all three. But what has happened is the pattern is disrupted. Do you see what I'm saying? The stripes are all messed up. Now, this particular forest officer, Debabrata Swain, he approached me and he said, can you figure it out? Because for him, it was simple-minded. Why are these tigers this way? Why do they look this way? And I kind of thought, well, it seems really difficult, but I had some other insights. There was already some work on color morphs in tigers. White tigers have a mutation in SLC45A2. Golden tigers potentially have a mutation in this gene called corin, though we have not figured out the mutation in the golden tiger in India yet. But overall, something was known about the genetics of color morphs. So this tiger has no orange, this one has no black. But what we had here was more of a pattern morph. And some beautiful work by some colleagues and collaborators at Stanford had shown that, when you go from this type of a patterned cat to this type of a blotched cat, there's mutations in this gene called TACPEP, which is a transmembrane peptidase. So mutations, different mutations in this TACPEP gene, just single mutations though, result in going from this phenotype to this. It's a developmental phenotype which controls striping pattern. Not only cats, but they also showed that cheetahs, this is the king cheetah, this is the normal cheetah, differences in the patterning, again mapped back to TACPEP. So since, of course, I jumped at the opportunity because it felt like exploring a phenotype in the wild, there's nothing more exciting than that, right? So basically, the question was, can we study this phenotype? Well, very difficult in Simlipal. Tigers are really, really rare. You see them once in maybe like a few months. But luckily for us, luckily for me, I had a fantastic PhD student. And also in Nandankanan Zoo, we had this litter that was born, which had, a black male, two normal, one male, one female, a white but black, like a pseudo-melanistic white female, and this is the mother, which is white. So this also shows you that the two traits, pseudo-melanism and this white or leucism, are independent, okay? So now we had a pedigree. I told you about the power of pedigrees, right? We could build a 3-generation pedigree. We could sequence genomes. We could do an association study to map whether mutations in TACPEP are likely associated with this phenotype. We got really lucky. It's basically a simple mutation which converts, it's a missense mutation in this peptidase M1 domain, which is generally very highly conserved across taxa, but is mutant in these pseudomelanistic tigers. And it's 200 times more likely to cause this phenotype, okay? So we potentially had a causal mutation from the zoo. Then we went back to the wild in 2018, 2019, surveyed tiger scats here because it's very difficult to see tigers, let alone sample them. Forget about the hair stuff here. We walked trails, collected fecal samples, extracted DNA, designed simple PCR approaches to look just for this mutation. And we figured out that yes, we had this mutation and its frequency was super high, almost 60%. Now, maybe we missed this mutation elsewhere. So, the other thing we looked at was Simlipal differentiate? This high frequency here and complete absence elsewhere is a signature of genetic drift or the fact that this population is isolated and small, right? So we investigated that. We identified only 12 individuals. Though it was not a population estimate suggesting the population size is very small. We looked at what's called population structure again, and we found Simlipal is differentiated from the close by Central Indian populations. And when we looked at pi hat or relatedness, individuals in Simlipal are almost like siblings. So almost 40% or so related to each other. Again, suggesting that it's a small population experiencing drift and potentially inbreeding. So basically, this is just explaining to you again what genetic drift is. When you have a small population, by chance, some mutations or alleles are going to increase in frequency. Like when you pull a ball, when you toss a coin just once, you'll get a head or a tail and you'll say it's a biased coin. But when you toss that coin 1,000,000 times, you're much more likely to get a 50% chance of heads and tails. So basically, we did simulations to explore how could we end up at the current allele frequency. And it turns out that the only way is if we had a very small number of founders. Again, we're coming back to founding effects. Similar effects in livestock, right? You have a few founding individuals, and these founders and their genetic makeup will then dominate the population for many, many generations to come. So in this case, potentially the founder must have had this mutation, right? And in fact, there was one tiger about, like 20 years ago, which was taken from Simlipal to the Nandankanan Zoo. And that individual must have also been a carrier. Otherwise, we couldn't have had it in the zoo population either, right? So we proposed to the department what's called genetic rescue, which is given that this population is really small, the genetics are very homogeneous, very low variation, high inbreeding, high and potentially increasing frequency of this mutation, the introduction of new genetic variation. And very much to our surprise, because sometimes you write papers and you talk to people, but nothing ever happens. The Odisha Forest Department took it to the National Tiger Conservation Authority, got approval, and we then suggested, then they came and asked us, okay, so you say we should move individuals, where should we move them from? And this was a bit of a difficult, like it's one thing to say, there's low variation, you need to augment it, but where do you get animals from? So we proposed this framework where we said, okay, you want to bring in diversity, new variation. You want to ensure there's no local adaptation. If for some reason Simlipal tigers have adaptations and you bring in some new individuals, they're not going to do well enough over there, right? And you also look for what's called genetic compatibility. So you look at the distribution of deleterious mutations, how much they overlap. If they overlap entirely, then even though you're bringing in variation, you're not going to offset inbreeding. Inbreeding will still continue because what was bad here is the same as what is bad which you're bringing in. So you want the least overlap of the bad and the highest good in some sense. Anyway, so we did these analysis and we proposed that Tadoba, which is quite far, is the best potential source of individuals to Simlipal. And what was really amazing is this actually happened. So in October 25, the Forest Department brought 2 individuals from Tadoba. They're very young females. They're still settling down. But it's going to be great to see once they breed, how this inbreeding, these runs of homozygosity are going to be broken up by recombination. Of course, we keep working. And what's really interesting is we've done fieldwork again in 2023, 2024, 2025. And basically, this allele frequency has now gone from 60% to almost 72% actually. So it's definitely increasing, and should there not be an introduction, it would get fixed. Now, I'm not claiming there's anything wrong with the black tigers. I don't know that. But it's definitely a telling sign of drift. We never had genomes from here because we were working with feces. But again, a fantastic graduate student named Abhijit has, of course, sampled shed hair, but he has also been able to sequence whole genomes, high depth whole genomes from feces. That's crazy, right? If you just take DNA from feces and you sequence it, 98% is microbes. Okay, so how do we get rid of that microbial part? We do enrichment, methylation-based enrichment, which like people use it to enrich the microbiome. We use the other part. We enrich the host. And when we do this, we are able to actually do genomes from feces. To the best of my knowledge, there's very few species this has been done in. And so, you know, the difficult permitting system actually pushes us to come up with these kinds of innovations. The other thing which we're doing, which is crazy difficult, is sampling historical material. This is a picture from a place called Akaltara in Chhattisgarh. And you can see there are so many trophies here. So for example, the Maharaja of Sarguja in Chhattisgarh has a hunting diary where he has documented the shooting of 2,000 tigers. Okay, these animals died. And remember, I told you I love animals. So I want to try and understand their story. What was their DNA? What was the past like? This is very challenging, as you know, because the minute an animal dies, the DNA starts degrading, right? So we have extremely fragmented DNA in these samples, but we have cajoled Maharajas, nagged them, troubled them enough, and we now have about 400 samples across India from the last 150 years. So it's been a massive effort, but already we see some patterns. For example, the samples collected from the British Museum, which is well temperature controlled, gives 1 nanogram per microliter DNA, whereas our trophies, sweating it out in Chhattisgarh, give only 0.2 nanogram per microliter. But we are now in the process of creating genomes with these samples. Now, all of them may not give us high depth and good coverage, but we'll have to see whether we can actually figure out not just what the variation in the past was like, but through these bottlenecks which have happened, again, drift and selection against deleterious mutations would have happened. So we can actually try to figure out what were the high fitness effect deleterious mutations. Okay, so this is a very quick slide, and please, you know, it's a new work. So sorry, I'll just go over it very quickly. I thought it would be nice to include since Subir is here. We have sequenced about 200 plus high depth lion genomes from across lion range. So this is, you can see, of course, there's lions in, oh, sorry, I'm going very fast. Sorry. Yeah. Across lion range. So there's, of course, we all know about lions in South Africa and all, but there are also very endangered populations in Central and West Africa. And for example, the Asian lions or the Gir lions are sister to these lions, okay? Now, these lions are almost extinct. Each of these populations has 50 individuals, so on and so forth, whereas 50, 100, 200, whereas we have now 800 plus Asian lions. But of course, when you look at how differentiated they are, you can see the PCA here compared to the earlier one. Remember, the tigers across the range, it was like 13%. Here, the Asian lions are standing out as 38% different. And the reason is because you can see they're all one on top of each other. Can you see that? They're all one on top of each other. What does that mean? Correlated allele frequencies, high inbreeding, low variation, right? And you can see that here. Basically, this is estimates of individual heterozygosity. And there's India, almost zero, almost no variation at all. This is the same plots of ROH. So remember, the lion genome is also about 2.4, 2.5 GB. So 83% of lion genomes are homozygous. This is nuts. This is crazy. I don't think, you know, we know of any species actually which has so little variation. So the question is, have we been wrong all this time? Have geneticists been wrong? And maybe you don't need genetic variation for survival. So I think this again becomes a really interesting case study to understand how much variation do you need. For example, whatever variation there is, where is it? What parts of the genome? Is there some complement of variation which is critical to survival? I should point out also that these lions are under relaxed selection. There's a lot of medication happening of the wild lions. They have succumbed in the past to diseases like CDV. So, you know, I'm not saying that they're home free, but definitely it's a very, very interesting case study and not yet published, hopefully in the next few months. We did some work on elephants, which showed something really surprising, that when you look at elephants from, say, the north to the south, you find what's called a serial colonization effect. That is, it looks like elephants came into India from the north, and as they went southward, they had less and less variation. Okay, so it's like you take some, and then you take some of this, and then you take some of this. That's exactly how human populations are. The largest amount of variation for humans is in Africa, and the further you go from Africa, the lower and lower the genetic variation is. What was interesting is these are really deep divergences, and this is really important for conservation because, for example, this population south of the Shenkota Gap is very endangered, maybe 50 or 100 individuals, and yet pretty much no conservation efforts are being focused here. Okay, sometimes we use genetics as a tool. I will try and quickly wrap up. For example, in the case of dholes, you can't identify them visually, they don't have stripes, so we can actually walk through the forest, collect samples, individually ID them, and do actual population estimates. Because, for example, we really had no idea how many dholes there are, just as we have no idea how many wolves there are, though we say wolves are endangered, right? So this was actually the first time we were able to do a statistically robust estimate of wolves. We use fecal samples also for things like understanding diet, so metabar coding. This is again work on dhole in Tadoba. This is Tadoba core, buffer, and territorial in 3 seasons, monsoon, winter, and summer. And we showed that, if you just take these pinches of the scat, you can actually do metabar coding and figure out diet. What's really interesting about dhole is, unlike, say, leopards, which are very plastic in their diet, dhole are extremely tight in their niche. They eat only wild prey most of the time, very low presence of cattle and so on. Okay, you keep hearing in the news about human-wildlife conflict. I don't know whether you guys saw recently that just yesterday or day before yesterday in Tadoba, 4 women were collecting tendu patta and they were killed by a tiger. So what do we do about this? We need to do something about it. There are some areas where tigers are abundant and they're in conflict with humans. So we did something what we call kill site investigation. This is a jugular bite mark. So when carnivores kill, they bite at the jugular. You can see this, these are teeth impressions. And we just took Johnson's earbud, like a swab, put it in there, and we were actually able to identify the tiger individual just based on the DNA, potentially from the saliva. We also sampled lick marks. Cats lick their prey. I don't know how many of you have cats. They really clean their prey, and also there's a lot of hair on the body. So basically, we were able to identify so many unique tigers. This is a study from Kanha and Bandhavgarh. And I'll just skip to these results just to show you that this is the distribution of, like, at how many kill sites was 1 individual found, 2 individuals, 3, and so on. If there are problem individuals, that is 1 problematic tiger going and killing everything, right, which is what the media gives you the impression, right, like Avni, right, then we should have had many points here, a tall bar here. What we have is a tall bar here, which means many tigers are killing once in a while cattle. Okay, so this means that it's almost become ecological. There is so many cattle, and cattle doesn't do much. It just sits down. When the tigers chase them, they just sit down. They don't run away, unlike deer. So it turns out that cattle is now becoming a disproportionate prey for tigers, especially even in tiger reserves. This is something which we need to think about. How do we solve this? There's also spatial pinch points where there's more conflict. And one approach might be, I mean, maybe cattle, you give up on cattle because, well, there's compensation and so on. But when there are human deaths, there has to be some action. And unfortunately, science is way too slow. Like I go, I sample, come back to the lab, wait for an Illumina run. It takes too long. If we could actually take a handheld nanopore to the field, ID the individual in the field, and have a database to compare it to, not just a genetic ID, but a physical ID, then we could actually bring this into action. That would be fantastic. I would love it if I could help that happen. So I'll just spend a few more minutes talking about our work as Dr. Gaurashankar also mentioned on zoonoses. We know that 60% of all diseases humans get are infectious. and 75% of them are emerging. So, you know, we are mammals. We're related to all other mammals. And so basically, these diseases spill over to us from mammals. There's various pathways for spillover, live animal markets, of which we have a lot in India. Let me tell you, they're all underground because legally hunting is not permitted in India. Okay, you cannot even hunt an ant legally. So these live animal markets are generally underground. There's a lot of wildlife hunting. That's what we are kind of working on. Of course, wild animal farming of like exotics, like ostriches and so on. And of course, lots and lots of domesticated animals. If you look at predictions about where we expect zoonoses to emerge, India is a hotspot. Why? Because it has high mammal biodiversity, high land use change, small parcels of fragmented land. Remember that image from 1700 India and India today? And of course, high human population density and high livestock density. So overall, this is an important question also because the frequency of novel zoonoses has increased, as has the economic cost, as has the geographic footprint. Because of commercialization, because of these networks of transport, a small isolated spillover event now has the potential to become a pandemic. So if we look at India itself, where are the mammals in India? So I'm sure you all know 2/3 of India's vertebrate diversity, mammal diversity is in the, where is it? In the northeast, right? So the northeast of India has a large, disproportionately large proportion of India's diversity. So you can see in red primates, large number of primates in the northeast. This is bats, rodents and shrews, and all of them together. So the northeast of India has high human, wildlife, livestock interface and density, and there's a high chance of spillover here. This is a picture of a bat harvest in a place in Nagaland. Basically, these people have smoked a cave, the Bomar. They're a group who traditionally performs these harvests. They've smoked a cave, and these are all dead bats, which they consume, right? So I'm not demonizing any traditional practices, but I'm saying that it would be important for us to study this to better understand the potential for zoonotic spillover in these contexts. For example, there's also a festival in Arunachal called Aran, where there's a large hunting of rodents and small mammals. And many diseases like scrub typhus, borrelia, etc., vector-borne as well, are from here. There's some very interesting customs as well. For example, the Mishmi have a custom where after they hunt, they spend 5 days away from their family in a separate room, almost like a quarantine, right? So it's very important for us to understand these things. And I've been working in the Northeast for 15 years, and I can tell you, I am very much a foreigner there. So it became very clear, at least to me at some point, that it's really important for us to do this work, but it must be done in a proper way with local partners. So we are actually working together with many institutes, and the goal is to basically try and understand unknown pathogens in Northeast India. Dr. Nagendra is on the program monitoring committee, and he just visited us a week ago or so. And we're trying to sequence potential pathogens from bats and rodents and livestock and humans, looking also at serology, because I think that's really important. A microbe is a co-occurring microbe, right? You don't know whether it's pathogenic until you look at serology. And we're collaborating here with Rahul Roy from IISc, who's developing multiplex serology methods. And then we're hoping to understand some patterns to get us to this kind of understanding. Hazard exists. Animals are around us, they live around us, they exist, they carry potential pathogens. Bats are amazing. They have an amazing immune system. They carry so many viruses and they don't get sick, right? That's hazard, that exists. We cannot negate that. On the other hand, exposure is what brings us at risk in some sense, right? So what we're trying to do is we're also doing a lot of community surveys and surveys of people to ask how much they interact with wildlife and so on and so forth, so that we can get an idea of risk. Vulnerability is, of course, where potentially all of you could come in. For example, if there's a potential pathogen which looks to be very infectious, we would have high vulnerability to it, and we could design a vaccine, for example, early on to reduce our vulnerability. Or populations which are very far from PHCs have high vulnerability. Okay, so this is just some pictures. This is actually a cave where Dr. Nagendra also came, and we catch bats and we have to work with them at night. We sample them, small mammals, of course, all with extreme biosafety. I want to be sure that we are practicing like extremely good, biosafety practices. And just a very quick visual to show you, for example, for these different different bat species, when we sequence just viral diversity, we find all kinds of viral families. And we're sure to find completely new families, known families, from neighboring locations like China and Vietnam and so on. But unfortunately, there's been almost no work of this kind in India. So it's going to be really useful. So in terms of, I think it's a really exciting, I mean, there's a lot of, I'm sure as students, you hear a lot of depressing things about the state of science and in India especially, or globally, and how things are not, could be better. And things can always be better, but it's also really exciting time. So for example, I think, and I'm a big fan of non-model, studying non-model systems. Just recently in the last 2 years, there's been these crazy discoveries, like studies on the naked mole rat, which have revealed the secrets of its longevity, okay? Or similarly, the bowhead whale and why it lives for so long. So these things can help us understand not just about the naked mole rat or this whale, but also about potentially how to improve human lives, right? So this wild animal non-model organism genomics, I think, is really a potential for the future. For example, those caves we went to have blind fish, okay? They just don't have eyes in Meghalaya. So I'm sure that studying these non-model systems can also give us some really, really interesting insights. We're living in the days of colossal, they recently have created this woolly mouse. I hope you guys have heard of Colossal. Paris Hilton supports them, so everybody would have heard of them. And I don't think they're doing de-extinction. They're creating new things. They haven't created a dire wolf. They have edited the genome of a wolf and created something new. So there's also a lot of ethical dilemmas which you all as young people will have to face. But I think the power of genomics, especially when combined with pedigrees, candidate gene approaches, and the power of also all the biomedical knowledge so far. We know so much from model systems. How does that translate into non-model systems? I think the future is really, really exciting. And I wish I was 20 years younger. There's a lot to explore. So I'll stop there. And I'm really lucky to work with fantastic people who take on all these risks, like sampling tigers when they could be around, kneeling into carcasses of cows, and bats and small mammals. So thank you so much. I'm happy to take any questions if there are. Yeah. Yeah. Yes, yes. Diversity. Yes. Yeah. Yeah, yeah, yeah. Yes.
So it's difficult to me, like because they are and he says like very best, right?
Yeah, yeah.
So, and all the action is not explained by the genome data. So
yeah. Yeah, yeah. So for example, that's something which, for example, with the lions, we're really thinking, for example, we have not looked at structural variants. We have not looked at epigenetics, right? This is just like Illumina genomes, yeah, like 15 to 20x, yeah. Yeah.
Contradicting biology.
Why? I'm sorry, what's contradictory?
Because, for example, like a PC.
Yes.
We are plotting like a taking it like a
yes.
first component PCA was
13%, yeah.
So that's we use it genetic.
Yes, yes.
Maximum variation explained by the genetic data is this.
No, no, no. Yeah, yeah. So the maximum in terms of ordination, right? Yes. So basically the most you can explain with a few dimensionality is that. So most of it is similar. That is, so whatever, 83% is the same. Yeah, 17% is different. Yeah.
In second
Second PCA. Yeah.
Yeah, you said like around 60 to 70%, 50% like have like a similarity.
You're talking about the lions?
Yeah. So
Yes,
that's
yes.
about,
Yes.
but only in Asian you have shown like some kind of like a mixing thing.
Oh, no, no, no. So in the lions, basically what's happening is, so when you have like when you have some variation, say you have some variation in South and East Africa, so it'll be what's like isolation by distance. So you'll see some spread of the points, right? But when everybody is the same, they'll kind of all be one on top of the other. So there's no spread of the points in that space as well. So basically they're so different because they're all the same as each other. That is reflected in the plot, right? Yeah.
Like if you have like 60, 70 similarity.
Yeah.
So it seems like a might be using this marker.
Might be using?
This marker to make this.
No, no, no. These are based on like millions of SNPs. So it's like whole genomes, right? So about 3 million SNPs. Yeah. 3 million SNPs. Yeah. So basically, I think what you're trying to indicate is that you could use something like an ancestry informative approach where you could use fewer markers and get the same differentiation. But our goal is to actually look at genome-wide variation, because when you use ancestry informative markers, you could do it so, for example, to establish, say, a breed of dog or something like that. But that doesn't tell you about the broader variation in the population or species. Yeah.
Radiation.
Yes.
So can we, I mean, for the 17%, the
Correct.
17% is the real temperature, right?
Yeah.
Because this is the variation.
Yeah.
Now, can we mark this 17%? Because this is what the percentage of the genome which is responsible for adaptability and for the survival.
Well, we don't know. All we know is that that is the only part there is variation in. So we need to look at it. Yeah.
Variation.
I know, but the thing is that maybe what right
So that's why 70%
is really important to understand. I agree. I totally agree. But what I'm saying is the thing about science is we always find surprises. So maybe that's what I was saying, that maybe epigenetics or, is able to contribute phenotypic variation, right? It must be contributing, and in the context of very low genetic variation, maybe epigenetics is compensating. I don't know. I haven't tested it, but maybe that is enough to give some more variation, right? I don't know. I haven't tested it. We know, for example, in Asian lions, we are seeing inbreeding phenotypes like ataxias, again, crossed eyes, stillbirths. So we're trying to look at stillbirths now and sequence stillbirths and see, do they have a different load? Because everybody is the same. So why some mate pairs have stillbirths and others don't, right? So I think there's still a lot we don't know. These are generalities. Yes, it's good to have variation. It's a very general statement, but I think we need to understand the specifics better.
Pure line mod.
No, there's nothing like pure line, right? Yeah. No. How? No.
Like pure line also, like you make it like a 70-80%. They are genetically pure like that. Like
But there's nothing like that here, right? What do you mean by pure? I mean, you mean ancestry?
80%.
Yes.
Pure lines also 70-80% like close to each other.
Yeah.
So, yeah, maybe you can refer any pure line model like.
Even the zoos, they do not have data on the pedigree. So.
No, other animals.
Which animals? I mean, every animal is different. No. Yeah. So what we are trying to do, though I didn't mention it, is we're trying to look at functional effect of some of the mutations by making cell lines of cats. But we've not been able to do that yet. Making cell lines for humans is very standard, but very difficult for anything else. So we're still trying. Yeah. Yes, 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 definitely. Yeah, it is not easy, but on the other hand, yeah, it is not easy. Yeah, definitely it's not easy. Yeah.
So you used word inbred.
Yes.
So normally what we know for inbred is in mouse is there and same, they are reproducing. But in the case of animals like tigers, there's natural selection going on already in the field, right?
Theoretically, yeah.
Yeah, no, it's going on because they have to fight for a mate, and then the pups has to survive in the nation.
Right.
So the one which we are looking, can't we say in a different perspective that these are the best markers or best genes they have? And although we are seeing same thing.
No, unless we test that, we cannot say that for sure. It could be by chance, right? It could be drift, which is causing certain overrepresentation, like for example, the black mutation. We looked for signatures of selection to look for whether the black is advantageous. It doesn't seem to be.
Yeah, so that's why I want to come to the question. The black is not advantageous because it will not camouflage to the environment. So it will be eliminated slowly. Since it is in zoo, it can survive, but in natural system, it won't survive.
This is a wild population I'm talking about.
How much is the?
70%.
70 are black.
Allele frequency is 70%.
No, in wild, how many are there?
In the wild.
70%.
Allele frequency in one population is 70%. So 50 to 55% of the tigers are black.
So in a specific location.
Yes.
So maybe that it's giving advantage to tiger for.
It might be, but unless we test it, we cannot say it is. Yeah.
Okay, thank you.
Yeah, very nice talk.
Yeah.
It's related to his question. So, are we giving too much of an importance to the depression part of inbreeding? Because inbreeding is also a method by which you will enhance certain traits. We only look at the homozygous
recessives,
minor homozygotes which come out, and but there may be a selective advantage these may be having. For example, cheetahs, I'm sure you would know, they have inbred for speed, but they have lost MHC diversity.
Yeah, the verdict is still out on that, but anyway, yeah.
Yeah, it's the hitchhiking effect that goes. But so are we giving too much importance to depression? But there is also.
So yeah, so that's the reason for looking at this historical material, because what you're talking about is a process called purging, where highly deleterious mutations are removed from the population, and those which survive following a bottleneck, following inbreeding, will be ones which are maybe advantageous or not deleterious.
No, what I mean is we are looking at only the recessive alleles, which may be deleterious, but there may be several advantages of the recessive alleles as well.
Right, but generally, if we look at the distribution of fitness effects, most mutations are deleterious. That has been shown with many species. Most mutations are deleterious. So the disproportionate number would be deleterious. There may be a few advantageous ones as well.
And how is
regardless, I think to test for selection, I think all of your questions are about selection. We need to have a test for it. The problem with evolutionary biology for a long time has been a lot of just so stories saying, oh, this is advantageous, oh, this is so beautiful because it's advantageous. But unless you test for it, you cannot state advantage.
For example, in cattle, we breed for certain traits, right?
Yeah.
So we breed for milk yield. So you generally see that the traits increase when you inbreed.
Sorry, what will, because selective breeding.
The traits gets concentrated. So you do get a hitchhiking effect and you'll get some deleterious effects as well.
Yes.
But there are a lot of advantages with inbreeding as well, is my
point. That is with selective breeding. I don't think in this, where you're having directed evolution, you're engineering it by selecting for specific traits. But in the context of the wild, I don't think there's any reason to assume that there would be selective amplification of increase of frequency of certain traits.
Okay, you're measuring fitness, right? So
yes.
How is fitness defined in the wild? How much?
Fitness is defined by lifetime reproductive success. Yeah, which we can't measure in these species. It's very hard. But what you're saying is right. In case of selective breeding, you might be selecting for certain traits, and there may be correlated traits which go along with that. Yeah. They're either linked, and especially, for example, in this case, when you have these wrong runs of homozygosity, you will have those traits which is in that homozygous run, which are coming along, right? But I'm not sure there's any reason to suppose that inbreeding here is happening in the context of enhancing certain traits. Great.
Ma'am, great talk.
Yeah.
My question is not related to what you spoke about. It is just a general question. So basically you have been to wild and you have a very good knowledge.
Yeah.
So my question is, what you are trying to address is how homozygosity can cause the variation and not variation, how it can
how it affects population.
It can affect the population. So this is one aspect. Ultimate aim is for the wildlife conservation. So what do you think are the other areas, like you have been studying in terrestrial mammals, there are aquatic mammals also. So what do you think in your perspective are the other gaps which need scientific focus studies? And suppose like I am from a diagnostic background.
Right.
So if I consider that, okay, going into a wildlife research, I consider myself as going into that field. So how I can be able to apply what I have gathered in my PhD into wildlife?
Sure.
And what are those actual areas?
So yeah, so basically, for example, like at least at this point, I think there's a lot of knowledge gap itself, right? So we don't really know a lot. But for example, once we know, for example, if we do this Northeast project and we figure out there are some potential viruses in bats which are potentially pathogenic, and we need to monitor their frequencies, then we can immediately diagnose like sentinel sites or monitoring protocols. So the minute, like with humans now, we have a lot of monitoring. We're monitoring for fever, we're monitoring for this, we're monitoring for that, what you do in the hospital, right? Diagnostic tests. We don't have those yet in the context of wildlife. And also, I think that livestock as an interface for wildlife, and for example, what Dr. Gauri Shankar brought up, AMR, something we're going to start working on now, is looking at AMR itself and the flow of AMR genes through these systems. Because you're just looking at the livestock. But I don't know, there's so many cows inside the forest. Okay, so there is an interface between livestock, dogs, humans, wildlife. And this interface is the most accentuated in India. I don't think there's any country in the world which has such an interface. So I think that one way is to open your mind to wildlife, right? That whatever diagnostics, whatever we do, can also involve wild species and to trace these interfaces. I hope that makes sense. Yeah, also eDNA is something which is coming up in a big way to monitor for biodiversity, to look for AMR, so environmental sampling. Right? So for example, even in some of these cave sites, we're putting like sheets on the ground and we're collecting like water, or whatever is there on the sheet the next day and sequencing it. So these environmental approaches, they seem like there's a lot of waste of money, but good pipelines to analyze those types of data. What are they telling you? What is an actual trend, right? You just have a lot of data, but does that actually reflect some change? Those are the kind of things which we really need to focus more attention on.
I have a couple of unrelated questions.
Yes.
One is, I couldn't help but notice that the lowest extinction happened to be birds. Is it because they are arboreal and migratory, or is it species diversity or something else?
I think that, birds generally, because they fly, they're able to adapt and go to novel habitats a little bit easier. But the probability of extinction is generally high for large birds. For example, frugivores like hornbills, which need large habitats for their sustenance. But since many birds are small and insectivorous, they're not so badly affected.
The second question is, has anybody studied looking at human population, for example, after a pandemic that basically wipes out a major population? I mean, 1918 influenza is one example. What do we learn from there?
Yes. So, for example, in response to that, there was a study with humans, and in fact, David Reich from Harvard has just published a study with 1,500 ancient human genomes, but they looked at the Black Death. Okay, and they looked at graves of people who died of the Black Death versus those who did not, and they were able to find variants in certain immune genes, not MHC. MHC is very difficult to work with in ancient samples because it's very repetitive, very difficult to work with in general. They were able to find in interleukins and other genes which are potentially responsible for immunity against Black Death. I mean, susceptible, made those people who died susceptible, and then they were actually able to test it functionally to see what is the effect of those mutations, right? So those are the kind of studies which we can do. And for example, right now there is a burning question. We just had I think about 2019, there was many lions that died of CDV. And just recently about 5-6 tigers have died of CDV in Kanha. So it would be really interesting to try and understand, like, what are there mutations which predispose you to it or not. Of course, like someone was saying, this is all really hard and you may be completely wrong, but it might be useful. So yeah, you can test for selection of specific things. The other thing which David Reich and team have just shown is the recurrent evolution of lactose tolerance in European populations. So with these thousands of ancient DNA samples, they're able to show that in time, the duplication of the lactose tolerance developed multiple times. So there was a very strong selection. For example, one could look at diseases like malaria, those kinds of things, right? When there's a very strong selective force, which is killing lots of people, then you can find those signatures of selection. Otherwise, selection is a little soft and if a lot of people are not dying from it, then it's unlikely to find those signatures of selection. Yeah, you see, basically it correlates. It correlates with the signature of a bottleneck, and selection will look like an expansion from a bottleneck because individuals who have that advantageous gene are going to increase. Yeah.
Can I?
Yes. Yeah.
So I had two questions. One is on the functional genomics that you were talking
Yes.
about. Now that the data is increasing,
Yes.
are there any scope of using the artificial intelligence to analyze, or is it too early?
No. So basically, I don't know about artificial intelligence, but definitely all these new programs like AlphaMissense and stuff, we are using them. Basically, we're using, like very liberal is something like, VP, variant effect predictor. So we're trying to basically use bioinformatic tools to predict effects of mutations, prioritize mutations which are supposed to be high effect, and make sure that they're in the wild also, and then test them in cell lines. But we've not been able to get to the cell line part yet. We've not been able to create cell lines yet.
Okay. My other question was about the shrinking land area which is available. So I think that is again a selection factor. And is it going to drive the inbreeding depression, or is it going to select the species?
So that's really interesting. One of the things which we know so little about is animal behavior actually. Like what makes individuals decide to move? Everyone is not moving. So there are some tigers known to move 1,000 kilometers also, and there are others which don't move much at all. So what is it that makes some individuals move and others not move? So there's a very interesting work which came out recently in Science where they did like microsecond recordings of these fish, and they showed that very early in life, just based on behavioral repertoire, they were able to predict longevity. So maybe we can actually predict behaviors by using this AI kind of stuff to look for patterns and predict which individuals may move, which may not. We don't know why. But there may be some risk-taking individuals, and we don't know much about behavior.
So much, very interesting talk.
Thanks.
I have a question.
Okay. Hi.
Actually, when we see in the wildlife, like there is a population which is increasing also.
Yes.
There is a population which is decreasing.
Right.
And what you have shown is a very interesting data.
Yeah.
In livestock, over the time, what we have observed that nutrition, metabolism, and reproduction has a lot to do with the next generation which is coming up. So, like mostly herbivores, I have seen that herbivores population is increasing in wildlife, carnivores is decreasing. So is there something which is there associated with the nutrition, the kind of food availability apart from the pay?
Yeah.
So for that, is there like you have something in your mind that you would like to look at from the conservation point of view, apart from genomics, that angle as well?
Yeah. So, basically nutrition, for example, is very closely tied to reproduction. So if you look at, for example, tigers in the Russian Far East, their home range size is 200 square kilometers because the prey density is so very low there, right? Whereas in Nagarhole, it's 20 square kilometers because there's so much of prey. Ultimately, a carnivore's population size is determined by the prey abundance, okay? So if the prey are higher, the carnivore numbers will increase. Now, the problem is carnivores do not see cattle as not prey. They are as much prey to the carnivores as are deer. And there's disproportionately, exponentially high numbers of cattle compared to the wild prey. So even if wild prey increase, because there are so many cattle, just numerically the carnivores will prefer the cattle. right? And definitely there's also a correlation between prey availability and litter size. So in parks where there's very high prey density, litter size can be 3 or sometimes even 4 cubs. Whereas in parks like in say Southeast Asia where the prey density is very, very low, in Malaysia, the litter size is 1. Okay, so definitely there are lots of these things which are impacting. I think that what the problem is, I'm not saying that there's a problem with tiger conservation. I think we have done a fantastic job. The government of India has done an amazing job in protecting tigers, and there has been tremendous tiger recovery in some landscapes. But large parts of Chhattisgarh, Jharkhand, Odisha, West Bengal are still empty of tigers. And ultimately, I'm not saying we should have more reserves, set aside more land, but multiple populations which allow dispersal and movement and connections is the way forward. Okay, thank you so much. Okay.
That if you now want to conserve
Yes.
Asiatic lions.
Yes.
So what is the easiest way out?
Yeah, so this is this still on YouTube? Okay. So basically, what we're going to model now is what's called genetic rescue in the context of those Central and West African lions. So basically, since they are sister, suppose they bring in variation. Not, of course, you don't bring a lion and drop it into Gir. You somehow allow them to mate and allow the variation to spread. I think that is one option to increase variation in general. That's like a more backup strategy. But I think targeted efforts to understand things like what makes Asian lions susceptible to CDV, right? And are there specific mutations associated with stillbirth, load, or these ataxias? So what we figured out with the ataxias is that if you do a video and you use a software like Deep Lab Cut, you can map the gait and you can actually score it. So we can actually look for simpler ways to monitor phenotypes. And when some detrimental phenotypes are increasing, we have to manage or remove them from the population. I mean, I don't know, maybe, I'm not sure this came across, but when you think about livestock, there's a lot of management, right? And when you think of wild, you think there's no management, the wild is wild. But today, I think we're in a situation where the wild also needs to be managed for it to be sustained. So that kind of intervention.
That's fine, but I think that a methodical kind of a planning and a SOP to conserve lion is very, very important. Whether you'll be successful in doing it or not, no one knows. For example, you have in a lot of zoos, you have lions.
Yes.
If you want, you can really do the at least artificial insemination you can do by detecting ovulation.
Yeah. So, yeah.
You can enforce a foreign gene, like you can take a staved sperm straws from other part of the world and try doing it. At least you will generate few males. But then you have to have a center in wild where you train them after birth. To be wild lions.
So basically, I mean, you're on the committee for CZA, I noticed. So we have a proposal with CZA also. So that is hopefully once we publish this, unfortunately, the way I work is first publication because it's peer reviewed and gold standard, right? So once we publish this, we will be talking about this with the CZA because even in the zoo community, lions are just dying, right? In zoos, they're just dying. Very young, they have abnormalities. So we can't ignore it, that it doesn't exist. So we need to find
the right place is to start from there because you have a nice research possible there. Which is not possible in the wild.
In the wild, correct.
And the data can be used as a baseline.
I totally agree. Yeah, that is the point of, so basically we're working with Etawah Safari Park and Zoo. That's where they have all the stillbirths and these ataxias. So we're standardizing our approaches from there. And then we want to go with, say, how do you set up a breeding program? What would you look for? Like you were asking, right? These mutations, when they increase, that's not a good sign for the population. How do you bring in new variation? How do you monitor that variation has come in? Those are all things which have to be set up, as you're saying, as SOPs.
And also try in case of livestock, probably generating more females is important. But in such cases, probably generating more male lions with a different genetic background is important because you throw them in jungle and they will impregnate many.
Right.
So I don't know if the same machine of XY sperm separation can work for lions from
Yeah.
other parts of the world.
Yeah.
So you try making more males.
So that we have to see those. We can do these
Keep
things.
those things.
Yeah, yeah. Thank you so much. Thank you.
Thank you, ma'am. Thank you so much.
Yeah, and thank you so much for the invitation.
So dear all, distinguished guests, colleagues and students, as we come close to drawing the curtain on the 15th Foundation Day lecture event of NIB, it is my privilege to place on record our deepest gratitude to all who made this occasion truly worthy of the milestone it marks. Begin with our respected director, Dr. Ravi Kumar Gandham, the vision and the steadying hand behind what NIB stands for. We thank you, sir, for your inspiring leadership in conceptualizing this event and the trust and freedom you extended to all of us in bringing it to life. Our heartfelt thanks to the Chief Guest, Dr. M. Ramesh, IPS Commissioner of Police, Hyderabad. Your distinguished presence lent this occasion both gravity and warmth, and reminded pursuit of knowledge and the society are, at best, one and the same endeavor. Our profound gratitude is due to our distinguished Foundation Day lecturer, Professor Uma Ramakrishnan, Madam. Your lecture was not merely a scientific address, it was an invitation to think more deeply, carefully, and to act more purposefully in the service of knowledge and conservation. Thank you so much, ma'am. We are honored by our distinguished guest of honor, Dr. J. Gauri Shankar, Dr. Subir S. Majumdar, Professor Ullas Kolthur Sitharam. Sir, you all bring a legacy of scientific excellence that elevates every forum you grace, and your presence today carries a special meaning. You have been a part of NIB's leadership and its journey. You have witnessed its institutions grow. You have helped shape it so. Your presence today has been a privilege for us at NIB. Warm regards to all the scientific colleagues for your presence. We thank all the heads, officers, and staff of administration, finance, stores and purchase, estate, engineering, housekeeping, and ICT teams. Thank you for your assistance and efforts. You are the backbone that holds everything together. A special word to our students. Your energy, curiosity, and enthusiasm are the very spirit of this Foundation Day. You are the future of NIB, its presence, and the finest investment that NIB is making. And to last, our Scientific Event Management Committee. What you see today is a visible face of the countless invisible hours, the efforts. So I thank the Scientific Event Management Committee for that. Lastly, if I have inadvertently missed anyone who has contributed in any measure, seen or unseen, please accept that this omission is mine alone, but the gratitude is whole. On behalf of BRIC-NIAB, thank you all. Thank you so much. One more thing. So we will also be having the games and student activities. We request you all to be here at 2:30. The lab visits will be carried out post lunch. So you are requested to be in your labs, please. 2:30. Maybe 2:30, please. Yes.